Welcome to Elena and Vivian's CRISPR project. Anyways, let's start. Explain the process of CRISPR and how it can affect the expression of traits in the future. What are the risks and benefits as well as the ethical concerns related to this new technology? DNA stands for deoxyribonucleic acid, is found in the nucleus of eukaryotic cells and is in the form of a double helix. The double helix consists of a backbone made out of phosphate, deoxyribose sugar, and nucleotides. Nucleotides are the building blocks for DNA. The nucleotides are adenine, thymine, guanine, and cytosine. In the base pair rule, adenine and guanine are the bigger bases, thymine and cytosine are the smaller bases. Adenine pairs with thymine using a double bond, guanine pairs with cytosine using a triple bond. The structure of DNA is also antiparallel, with half of the strand running the opposite way or being upside down. Each base can either be a template or a complementary one. Central dogma, also known as protein synthesis, has two major steps: transcription and translation. Transcription happens in the nucleus. In transcription, RNA polymerase cuts the DNA and glues nucleotides to create an mRNA, messenger RNA strand, which moves out of the nucleus to the ribosome in the cytoplasm. In the cytoplasm, more specifically the ribosome, translation occurs. The tRNA transfer RNA that holds the amino acids and anticodons attaches to the mRNA strand that has codons. The amino acids come together to create a chain connected by peptide bonds, which eventually forms a sequence that becomes the shape of the resulting protein. The protein's function depends on the order and length of the amino acid chain. Mutations occurs when anything changes in the original DNA strand. There are many types of mutations, but the most prominent ones are substitution mutations, frameshift mutations, and nonsense mutations. Substitutions are when a base in the DNA sequence is replaced with another base. Most times, substitution mutations are silent ones, meaning it causes either harm nor benefit. Frameshift mutations are either addition or deletions, where it adds a new base or deletes one that makes the whole sequence shift left or right. Nonsense mutations are when a stop codon occurs early in the DNA sequence, causing it to be premature. Only substitution, frameshift, and nonsense mutations cause change in the protein. Silent mutations don't affect the protein at all. Substitution, nonsense, and silent mutations don't shift the DNA sequence while frameshift mutations do. An example of a mutation will be cystic fibrosis, which is caused by 3 deletions in the DNA sequence in the CFTR gene. Cystic fibrosis causes abnormal mucus builds up in the lungs, which leads to lung failure and difficulty breathing. Another example would be lactose tolerance, where usually a single substitution mutation occurs in the LCT gene, allowing the individual to continue being able to consume dairy products because it can still produce lactase to break down lactose. This also goes to show that not all mutations are bad, some are actually beneficial, and most are neutral, which is contrary to popular belief. CRISPR stands for clustered, regularly interspaced, short palindromic repeats, a tool that allows scientists to modify, delete, or insert new DNA sequence. CRISPR can be used to change the attributes of people, such as hair color or eye color. It can also be used to treat diseases like HIV, cancer, cystic fibrosis, etc. In CRISPR, guide RNA is used to target the mistake in the DNA sequence. To use CRISPR-Cas9, scientists first identify the genome that causes a health problem. Guide RNA will then be created and will be attached to Cas9. It will then move to the target cells. It locates the target sequence by finding the one that matches the guide RNA and cuts the DNA. This allows scientists to modify, insert, or delete new DNA sequences.
What are some possible applications of CRISPR? CRISPR can be used in many ways that may benefit us or may put us at risk. What makes CRISPR stand out and be so revolutionary is that it saves scientists a lot of time, money, and energy because it is so easy to use and has a high accuracy rate. A prominent benefit of CRISPR is that it can potentially cure cancer, a very serious disease that currently has no cure. Cancer results from a type of substitution mutation called missense, from frameshift mutations, or from nonsense ones. With the help of CRISPR, however, these mutations in the DNA sequence can be corrected to get rid of cancer in an individual's body. Not only can CRISPR help cancer, but CRISPR can also be used in a similar way to treat other diseases such as cystic fibrosis, Huntington's disease, and retroviruses like HIV. In the light of the pandemic right now, CRISPR might even be able to resolve COVID-19. CRISPR can also be used to enhance different traits of humans that will help them throughout their life. Such traits include intelligence, speed, eyesight, hearing, and hair health. CRISPR can give an embryo 20/20 vision, can prevent the embryo from ever balding in their life, increase their intelligence to excel in education and career, give them perfect pitch so they can be musically inclined, and make them a sprinter that can shine in running and perhaps cross country. In addition to these, other traits can be enhanced, such as strength. Another benefit of CRISPR would be GMOs, or genetically modified organisms. CRISPR can be used to change crops' genetic code to produce healthier and tastier foods. Not only that, but it can also help with prolonging food life/storage, drought, herbicide, and insecticide resistance, and overall improve the quality and safety of crops and agriculture. Now, CRISPR isn't entirely sunshines and rainbows. Benefits always come with risks. Such a risk would be using CRISPR with malevolent intentions. CRISPR can easily be in the wrong hands, being used for the creation of modified humans or superhumans or soldiers, for example. These superhumans can potentially destroy humankind and or take over society because of how strong and intelligent they are. Other evil intentions could be the creation of incurable viruses, even with CRISPR, such as a flu virus that's immune to the flu shot, and bioterrorism and biowarfare. Another risk of CRISPR would be reduced genetic diversity. CRISPR can be used to alter embryos' appearances so everyone might eventually all look the same. Take these 3 girls, for example. If they didn't wear different clothes or style their hair differently, you would think they were triplets or the same person. The world would become so boring. What's the fun in having no differences? A third risk of CRISPR would be unwanted effects. When CRISPR is being used to treat a certain mutation, mistakes can occur which will lead to other unintended parts of the DNA to be changed. These changes can come into effect right away or in a few days, weeks, months, or even years as the individual gets older. The ethics of genetic engineering. What are the arguments in favor and against genetic engineering of humans? There are many ethical concerns revolving around genetic engineering. Some are in favor of it, others are against it. Both sides have their respective and understandable reasons. Let's take a look at both. Genetic engineering provides children a chance of living without an illness, disability, or disorder. Roughly 65% of the human population are affected by inherited diseases. Most of these are incurable as of now. Genetic engineering can get rid of these diseases in embryos. For example, someone diagnosed with cystic fibrosis will be substantially affected throughout their life negatively. Breathing, a major part of living, would be a big struggle. Eventually, they'll die of lung failure or suffocation. However, the mutation that is causing this can be modified with genetic engineering to be removed, allowing the individual to live normally and have a longer lifespan. Genetic engineering can also produce matching donors for people with illnesses. For example, a child in the family is ill and is in need of a certain transplant. The second child that the parents plan to have can be genetically engineered to become a matching donor to help the first child. The second child can be free of diseases and the first child can be treated. A win-win situation. Genetic engineering can prevent diseases or disorders from being passed down to future generations, eventually eradicating genetic diseases. For example, a married couple both have Tay-Sachs disorder, which slowly eats up the brain and leads to early death. If not for genetic engineering, their chances of passing this disorder down to their children, grandchildren, and future generations are extremely high. Genetic engineering can remove this mutation from their children, which will obstruct the disorder from being passed down to subsequent descendants. An unethical thing about genetic engineering is that it can lead to a world that's genetically discriminated and segregated. Meaning, it can lead to segregation once again, like white people versus black people, but genetically modified people versus those who aren't genetically modified instead. Those who are genetically engineered would be favored, and naturally born people would be discriminated against. It would create a social ranking with those who are genetically engineered at the top and those who are not at the bottom. This is because genetically engineered people are viewed as more capable and of a higher caliber, therefore being more valued. A great example of this is in the 1997 sci-fi movie Gattaca. The non-engineered are considered invalid or unworthy, and the engineered are considered valid. They are eligible to work at Gattaca. A second argument against genetic engineering would be that it can lead to mental issues in people, especially children. It can cause low self-esteem and confidence and a sense of inferiority. For example, in a family with a first child that was naturally birthed and a second child that was genetically engineered, the first child might feel inferior to their sibling because they're not as competent as them. This can lead to self-doubt and eventually possible mental illnesses. The parents favoring the second child can add on to this as well. A third argument against genetic engineering would be that it goes against some religions. Some religions believe that their God is the one determining certain traits, that all things happen for a reason. Modifying or editing genes would be interfering with this natural system and would be violating their worships. For example, someone could have blue eyes, but we use genetic engineering to change them to green eyes. Essentially, this make believers in God mad because we challenge God's choices and alter them. I think we should be able to genetically modify humans to the extent of purely for treating inherited diseases and for altering people's appearances, eye color, hair color, etc. In the case of inherited diseases, the criteria should be that the disease is serious, where it will affect the individual in their daily life, will lead to discrimination or bullying by other people, or will shorten life expectancy or is deadly. Regarding appearances, it should only be for adults in order to avoid unrealistic and unfair expectations and standards. I think parents, doctors, and lawyers should decide and regulate what it is used for, because it is the parent's child, so they are responsible for them. Doctors know what is best medically, as they are the medical professionals and experts, and lawyers can keep their uses in check so nothing becomes illegal and immoral.
I think we should be able to genetically modify humans. Genetic engineering might make a large impact on the humane society and cause lots of benefits. For example, it can edit embryos' genetic information to prevent inherited diseases that may affect the health of humans, to have the potential of a longer lifetime, etc. It should be to the extent where it can help the humane society, but not to where it can cause harm. The criteria should be if the inherited disease can affect the individual's daily life and health or cause any inconvenience. I think parents should be the one who regulates because it's their child. Doctors should also be the one who regulate because they know what is best for the embryos.
To summarize, CRISPR is a tool that allows scientists to modify, delete, or insert new DNA sequences to prevent diseases and mutations. In the process of CRISPR, guiRNA and Cas9 enzyme work together to edit the mutated gene. guiRNA recognizes the mutation and Cas9 cuts the DNA for scientists to edit. CRISPR can change different attributes of people, such as changing hair color and eye color. It can also potentially end genetic diseases in the future, so more people can be cancer-free, for example. Although the benefits of CRISPR are significant, such as curing diseases like cancer and HIV and GMOs, the risks of CRISPR are considerable as well, including malicious use of it. Some people are in favor of genetic engineering because of its ability to prevent diseases in individuals and from being passed down, and to help other people who are ill. Others are against genetic engineering because of the discrimination and favoritism it can cause, along with the violation of religious beliefs. So, what do you think about genetic engineering? Let us know in the comments. That's it for today. Bye!